right. Over to you, Thomas.
Thanks a lot, Phil. Can you share in the Slack the link to Deposit Cloud
Yep.
for the
I'm going to
users?
do all of that. You got it.
Okay.
Just maybe lecture or talk a little bit while everybody's getting onboarded, because it takes everybody about 3 or 4 minutes to get into Deposit Cloud. So.
What I was planning. So thank you everyone for joining. I'm Thomas. I'm research associate at Harvard Medical School in the Department of Biomedical Informatics. So it's the CLS lab, Translational Data Science. We mainly analyze EHR, electronic health records. So we work with a few big hospitals in Massachusetts, MGB, Massachusetts General Hospital, and Massachusetts General Brigham, which is a consortium with also Brigham Women's Hospital. We also work a bit with Veterans Affairs. And so on. This is our laboratory website. One of the main pages is the software page. Here you can find a lot of the stuff we developed. Nile will be something that will replace for unstructured text data. You will have a dictionary with a code identifier and a label, and it will replace the words and the parts of sentences. It can be several words with a unique code. So it will take most of the time out all the stop words and focus only on the concepts you want. So Nile is an important software we are using. MAP is something to do phenotyping based on electronic health records. So electronic health records are filled up when a patient visits the hospital, and sometimes he may have a diagnosis of rheumatoid arthritis, for example, but if he only had one in his whole life, we might not really consider him a patient of a true case of rheumatoid arthritis. So MAP does this kind of modeling. And one of the important thing we'll talk about is Kesser, but we'll come back to it a bit later. I want to give a bit more about my background. So now I'm more in biomedical medicine and hospitals, but I did my PhD more in a drug discovery setting. I was at the Quartz Bio company, spin-off of Merck Serono, and I was working especially on a European research project, precise SADs, systemic autoimmune diseases. But my colleagues, the founders, were much more on the drug discovery side, and I was more on the research project. So during my PhD, I did genome-wide clustering, so both with candidate SNPs or genome-wide. The idea is like for systemic autoimmune diseases, it's a disease that have overlap in symptoms and difficult diagnosis. So the idea was to reclassify based on molecular features such as genetics, SNPs, RNA-seq. So this is the first package I published on CRAN. It's useful for GWAS kind of analysis. This is the usual linkage disequilibrium plot. A lot of people, I guess, are familiar with it. And I wanted to push this plot a bit further to have higher understanding of correlation structures in the genome. And so this is, for example, with hundreds of SNPs. So I invite you to check this package out also. This is the second package I developed to do density-based clustering. In this one, so here we have a classic example of density-based clustering where the clustering can detect shapes. And we were particularly interested in it because genetic signatures can be very noisy and we can't get a very good separation. So we wanted to focus on core clusters. And so in this example is a DB-scan algorithm. This shows a limitation of DB-scan when you have different densities of clusters. On the first above side, it's a DB scan with a specific threshold. He detects the cluster on the left and on the right. But on the right, there's two inner clusters. And on the lower figure, it's with another threshold. And in that one, he detects the inner clusters, but not the left and the right. So this is the limitation of DB scan. So an existing method for that is optics. And I did a little algorithm heuristic to derive clusters from optics. This is the reachability plot from optics. On the left side, we have the left cluster, on the right side, the right cluster, and these two valleys are the inner clusters. And so optics enables us to see them, and the method I developed, optics KXI, enables us to really identify them automatically. So here are a few more examples on example data sets, and this is what it would look like with more real world data, which is a bit harder. You don't really know how much clusters there are in. And so my package provides a distance-based metric, and you can compare a few clustering models to see what looks the best for your data. So this was a bit my background, my PhD. After that, I did 2 years of computational linguistics, trying to train myself into this new field that was developing. So I did that for 2 years as an independent. And after that, I did 3 years of developing a Shiny app for real estate analysis, so geostatistics. And so that's a bit the background. And so today I'm going to talk about the work I started with the CLS lab in January. So here is a good example to get into it. This is the kind of knowledge graph that we are working on. Knowledge graphs can have a lot of different definitions. One of the common one is that your edges in your graph will have specific meanings. So for example, you can have a graph with disease, drugs, and laboratory tests. And so one edge could be may treat. So from a drug to a disease, one drug may treat a disease, and a laboratory test may be associated with a disease. So we would have two different edges associated with and may treat. So this is the kind of a common sense definition of knowledge graphs. And in our case, we're more, we have similar types of edges, but they are proportional to the association power. So, for example, if you have SNP data with a disease, you will have a p-value association, and in here we would have a higher node, a larger node for stronger associations, and the edges would be smaller as the association is stronger. So the p-value example is one. It's another app that we're working on that I can't show you today, but that will be public a bit later on. In this app, we're doing natural language processing. So it's terms labels. Like for example, eating disorder is 2 words, but it's one medical concept. And so we train our embeddings using Word2Vec on a large unstructured text dataset. And it gives us similarities between two pairs of concepts. So for example, here, eating disorder, suicide or self-inflicted injury. And so we have a pairwise cosine similarity matrix between all our medical concepts. And the knowledge graph will enable us to show which are strongly associated with both our target nodes. So here our target nodes are suicide and eating disorder, and we map out all the concepts significantly associated, And so on the upper side, we have those associated only with sleep eating disorder. And in the middle, we have those associated with both. So as we zoom in, we can get more information. The label, the legend doesn't show up here, but so just to show you in, green will be medications, red is laboratory tests, blue is diagnosis. And so in this app, we can click on a node to see children node. So for example, antipsychotic medication, if we click on it, we have all the different medication. So this is an example of the knowledge graph we are developing, and the K-graph package I published a few months ago does these kind of graph. And this is what we're going to go through today.
Thomas, do you mind, quick question that's coming up to me in the chat box. Do you mind putting the link to this in the Zoom chat if you have that? And then also if you have any slides or GitHub repo, if you could put that in the chat too. And then I'm happy to redistribute that during the workshop.
Yeah, so there are a few links in the description. There's in the description of the workshop, there's the link to the
Perfect.
K package. There's a link to the GitLab repo, but I can also.
I can get those. That's perfect. I didn't realize they were there. So yeah, that's perfect.
Yeah, so I don't really have slides, but I have. This is a study I did on specifically on mental health that I will come back a bit later on. My main support will be the knowledge graph vignette. So the app I developed for our lab enables us to produce the similar graph as this, but with some data sets that anyone can upload. So these links are publicly available. You can use them if they are if they have too much visitors, like if it hogs too much our server, we might close it down, but for now it's publicly available. And so we can input concept relations data frame or embedding mattress or co-occurrence, and I will explain a bit more via the vignette. So in the background, I explain a bit what I said previously on the knowledge graphs. So sets of nodes in relationships. Nodes and edges can be of different type. One commonly used of relation is parent-child. In the context of mental health, it could be schizophrenia is a mental health disorder. The sgraph package is a companion package that focuses on creating the HTML widget. The underlying visualization is in JavaScript. So the sgraph package just does the HTML widget to the Sigma.js library, and the kgraph package calls it. So in this vignette, we'll mainly see building a knowledge graph with specified relationships, then with determining a similarity threshold for embedding metrics and measuring the performance of the prediction model. So give me a second. So in the GWAS catalog website, we can download some summary results from existing previous genome-wide association studies. And the example I took in the vignette is for my main research focus at the CLS lab, suicidal ideation and suicidality in general. So this is this EFO 7623. And I was thinking that in this workshop, we could try to, that the participants can download any data set from GWAS catalog concerning their disease or disorder of interest and try to do the similar visualization as I do in the vignette. So here is the button, download associations. And so you can search for maybe rheumatoid arthritis. Download the associations. And it's basically this file that we will feed to the kgraph functions. So I'm going to first walk you a bit through, explain to you how it works. And I think that after that, we can do a little pause. I'll let you try out on your data sets of choices, and we can come back to it. So in the example in the vignette, I already pre-processed the CSV file. The input we're going to want to give to the kgraph functions will be 2 columns defining the concepts in relation and the strengths of the association in the weight column. And so in this case, it's minus log 10 p-values. So if we open up So if we open up a CSV file downloaded from GWAS catalog, it looks a bit like this. So we have to identify the columns we are interested in. So in our case, we will have first column will be rheumatoid arthritis. Second column will be the SNPs. So there's one column disease trait over here. So it can be a bit different, like several kinds of rheumatoid arthritis. And I saw the SNP column somewhere. Here we have SNP gene IDs. Oh, here we have the RSIDs. So you would build concept 1, trait disease, concept 2, SNPs, and concept 3, weight columns, sorry, p-value, and you would transform then previously to minus log 10. So in my kgraph package, I have a little script that does that. But in the vignette, I just load directly the pre-processed RDS file. So just to give you a bit of heads up on what we're going to do, once we have this data frame, we can call the build kgraph function. We give it our node of interest. So in here, it's not disease trait, it's just the EFO identifier. So 7623 is the one I downloaded, but there are a few others that are related. Maybe it can be a GWAS study that studied several types, maybe suicidal ideation, suicide attempts. So we will give it the concept we want to focus on and the data frame. This is the kgraph object that we can call with the sgraph package. So here I show a bit how I decompose the steps, but there is also some functions to call them all together. So once we have the kgraph object, call it with sgraph, we first convert it to an iGraph object, which is a very popular graph library. So this is just a convenience function, and this is the main function from the sgraph package. You will define a few parameters, node size, the label. You can just leave it as is. There is also a vignette of the sgraph package that goes a bit more into detail about the sgraph parameters. One important thing will be the layout. Or you can use all the layouts available in iGraph. And this is what it would look like with your node of interest and the closest associations. So here I have a dark seam, so we don't really see well, but we can see that it's kind of written over. So I normally develop it for it to be used in production in a light seam, but for development, I prefer to use a dark seam. And so when you hover above a node, it also puts up a frame. And so one drawback of the vignette, I especially developed this for a Shiny app. And in the Shiny app, it breaks down by line. So like the first line, you will have RSID. Second line, you will have cosine similarity. So in the vignette, it just puts everything on the same line because I do that in JavaScript. So this is the example with one target node, and we can also put two target nodes, and it would do a similar graph as this one. So this is the very basic call. And after that, we can map some information with a dictionary data frame. So in the dictionary data frame, we'll have column ID that corresponds to the concepts we defined in our first data frame. Desk for description, column, that will be the label, and we can put group and color. So it would look like this. Here, we don't have the EFO IDs anymore. We directly have descriptions. The color is, of course, self-explanatory, but the group is a bit so the thing is that it will try to pull together SNPs from the same group. So here we have a gene, and it will basically over this side, you will have all the SNPs that come from this gene. And so if you click on it, highlighted are the nodes that are connected to it. So this is a bit how groups work. So in this example, I just threw it together a bit quickly, but just to show you a bit how what the groups can look like if they are a bit more sophisticated. In this example, we have, so I can go back to the, in this example, the groups are a bit more meaningful. Here we're doing NLP. We have two suicidality target nodes, and the groups correspond to the family of concepts. So it can be disease, mental disorder. And so on this side, around this mental disorder group, you will have all the nodes that correspond to it. And on this side, you will have all the more general clinical concepts. So this is how the groups work. And so in the KGraph app, we basically have a package. We basically have two ways of doing groups. This one, which pulls together the nodes, and this one where you can click and have children nodes. So depending on your application. So in this first example, the groups aren't so great, but it was just to demonstrate how you could use it with a data frame. So I think maybe it could be a good time to maybe answer some of your questions. I'm going to have a look at the chat. I can't manage to see the chat. Okay, I can see how do we load the project, but I think there was maybe more questions, but I didn't see them. So don't hesitate to put them back up. So to load the project, you have to connect to the Posit Cloud workspace. So that's one way. Another way is just to install the kgraph package in your R console. And in the description, I also provided the link to this repository. And in this one, there's a Docker image for those who want to. And in this Docker image, you already have the kgraph package installed and a few others I developed. So should we try this out? Yeah, so literature data mining is the second part of the vignette. In the first part, we use only like pre-specified real relationships, but you could also do the same data frame with cosine similarities, for example. The in the second part of the package, we will see how we can do the knowledge graph based only upon the pairwise cosine similarity matrix. So this could be derived either via Word2Vec or with LLM embeddings. And another approach that is very similar to Word2Vec is to do co-occurrence matrix, pointwise mutual information, and singular value decomposition. So the knowledge graph for clinical trial data is this first example with, so it depends a bit what you mean about clinical trial data, but I find it especially useful for meta-analysis, where you can have several GWAS, results from several GWAS, and you can put it together to see which SNPs are shared between several phenotypes. You could combine several files from GWAS catalog, do a first data frame with one CSV file, or bind it with a second CSV file, and then put the two different phenotypes as target nodes and see what comes in between, a bit similar to here. So we'd have one example we are using in the Azure project is obesity, type 2 diabetes, and BMI. I can show you. So this is the repository of the kgraph package. And here we have a few functions I did just for the example data frames. The one thing I stumbled upon in the downloading the CSV file from GWAS catalog was that we could have multiple traits in the same cell. So I just did a little. That's basically the only pre-processing I do. I just reshape, put the multiple traits in several rows. So this might be useful if you want to try out another CSV file. Maybe in other CSV file, there are other issues you will encounter. So I'm going to take a 5 to 10 minute break to let you try out downloading a CSV file and calling the functions. And so you can ask me any questions in the meantime. I'm still here. Approach is really versatile in the sense that it can be used both for clinical trial data and literature data mining. In our lab, we especially do EHR text mining, but in this repository, I talk about one analysis on open access publications. I can try to show you what it would look like for the rheumatoid arthritis. Can we download a file? Can we download a file in Posit Cloud? So to answer the specific question, the rephrased question, it will be especially useful for numeric variables. So you can do adverse events, but you would have to do probably putting your weight as one. Maybe you can have sort of an association score for your adverse events. So you could have concept one, your medications, concept two, your adverse events. The weight could be the frequency. And so you could target two two drugs and see which are the adverse events that are shared.
Hey, Thomas, I have a quick question. People are asking about navigating the code in the cloud space. For what you've been showing, is the code in one of the folders, like the test folder or vignettes?
So in the posit cloud with kgraph like this, you should have everything.
Okay, if you go to files and files, plots, packages over to the right. Yep, straight down from there.
Yeah.
Yeah, right here. Yeah, so is the file, is there a file here that they need to run or work with, or or are they just going to be running the code directly in the console?
So how would we do if we wanted to download a new CSV file? Could we? Or
Yeah, I mean, so are you asking like to
so
put it inside a Posit Cloud?
yeah.
Yeah. So probably a couple different options. I think since people cloned the projects, it may, I don't know if they'll pick it up. So probably if it's a public data set, the best thing you could do is just put a link to it in the chat box. And then we can show people how to upload it using the RStudio environment. So if you have a data set, what you can do, see if you go straight down into the files pane, there's an upload button right there in the middle. It says new folder. Yeah. So you can browse to a local CSV here and load that into the Posit Cloud space. Yeah, just like that. So if you have that data set that people can access, you could share it in the chat box if it's not very big, or if there's a link to it, then people could get it here. And if you click on that data set, I think you can load it. Yeah, try clicking on that. Looks like it's a TSV file. So you may just need to have a separate bit of R code that will import that for you.
Yeah.
Yeah.
Yeah, so that's exactly what I wanted. Because so here I can show a bit what it would look like, the pre-processing of the file.
Yeah.
So I would do it, we can read lines or read CSV, the file, since it's a TSV, we'll put separator as \t. So here we read in our file, can look that everything went well. This is what it looks like. So as I was saying, what we're going to want to do is to create a new data frame with disease trait, SNPs, and p-value. So disease trait is this one. and we'll want to transform the p-values. Oh, there's actually already one that is transformed. Let me just check if it looks all right. Yeah. So we can use this one directly. So we just want to check that if we have some multiple traits, but it seems kind of okay. So this is our head. We can check our dimensions. Here's 3,800 rows. So now we're going to select one, which will be our target node. Come back to this function. So build kgraph. It's the new data frame I created. But it doesn't work. Let me maybe we can try the EFO ID. Yeah, so here we have the EFO IDs. So this is the what I was talking about, the fact that there are multiple traits. Here we see the first trait, second trait corresponds here. Rheumatoid arthritis, first trait. This one, the second trait. So we can give a try to the function I was talking about that I developed for the vignette example. So let's try to first recreate our kgraph data frame. This one will take, this time we'll take the EFO IDs. We give this to the reshape function. So it's a hidden, I think, or no, it's not hidden, it's exported. So it is hidden. Seem to have worked correctly. We should have a higher dimension. But now if we try again, okay. So just wanted to check something. Yeah, okay. So this time we computed the kgraph object with the built kgraph function. We first transform it to an igraph object. And then call the sgraph package on that. And here is our new visualization. So our target node is around in the center, this one, and these are all the strongest associations. And so we're going to do a dictionary data frame. So similar to what I presented here. So this time we'll have, for each row, the data frame will contain both phenotypes and SNPs. And so in this example, we have the EFO ID, which is with its phenotype textual description. And we have SNP IDs with their genes as groups. And we color by the type. So the dictionary data frame, we need to do something like prepare our phenotype data frame. We'll take yeah, this was our initial data frame. And this one with the reshaped. So what we can do is we'll first do the transformation. Okay, so we first need to define our dictionary data frame before we will reshape the multiple traits. So we'll want to do just trying to think about how I did it in the first place. Yeah, so our descriptions will be, we'll take the two columns, map trait and map trait URI. We can do a unique on this. And give it directly So here is our dictionary of just the phenotypes. And so to combine it, we're going to write it so we want to combine it also with the SNP dictionary. So the SNP dictionary will be I think it's map gene. We are bind the both. We call the reshape, make sure manage the multiple traits. Yeah, so this is probably because I expect that there is also a group column. I want to do that in 2 steps, but let's do that directly. So in our groups, we'll just copy the phenotype description and put the genes as the groups. So here we can see we have a different ID for the same description. Oh no, it's negative and positive. Okay, that's why. So we have our dictionary all set. And we're going to call it with the K-graph, the build K-graph. And this time I show the wrapper function where we don't need to do an intermediary iGraph object. This one, we add our dictionary. And we should be able to call this function directly on it. Here we go. So this time we have all the nodes around the group nodes. So this time our SNPs are put together by their respective genes. So if we have a look at HLA-DRB1, the SNPs will be over here. DRB9, they will be over here. So with only one target node, it's not so useful, depending on your use case, but let's try to put up 2 target nodes. So this time our second target node is over here. So we don't have a lot of association with it. So it's maybe better to have a more comparable. So we can have a look at joint damage measurement maybe. This is more the kind of groups we're expecting. And so over here we have our second target node. Here our first one. So we can find the SNPs that lie in between. Here I put one of the default behavior is to put all nodes that have only groups that have only one connection, or by default, those that don't have any group as other. And there's a lot of SNPs that don't have mapped genes. So I guess that's what's going on. So that's why we have a huge other group. It can be useful for maybe sometimes, let's imagine this group, it has some SNPs from the group associated with one and some from the other. So there's no SNPs really in common between the both, but the gene is in common. So this is, I think we can end the demonstration of the first part here. I'm going to have a look for the questions.
If you have any questions, feel free to post those in the Zoom chat or feel free to go off mute and ask it out loud. Either way works for us.
So the SNPs, basically, you said like there was the GWAS. Is it possible for you to go back there and guide us, like the SNPs for every type of disease?
The SNPs for every type of disease? What do you mean?
Like the genes that are associated with every like type. Is that how you derive that? Like you went to the GWAS.
Yeah.
So how? Oh, what can you elaborate on this, please, a little bit more?
Yeah. So how I use it is that I select a trait, a disease that we want to study. We can type it in here. So another disease could be lupus. So here we have several types. So we can select one. And here we will have a lot of what the results are reported in publications. So it's already sort of meta-analysis. It's all the significant SNPs that were found associated with this disease. And so some can have been performed on larger samples. So once we're at this page, on the disease page, we can click here, download associations. And this is how we obtain the CSV file that we transform further on. Does this answer your question?
Yes, thank you so much. And also, basically, like, is this like coming from like PubMed, or is this related to PubMed, or what is it?
So I'm not sure how GWAS catalog builds its database. It's possible that it's people who enter it themselves. So for example, here there was this submit tab. So the GWAS catalog team invites you to submit summary statistics of your genome-wide association study. They define the format, bit of documentation. And so the GWAS catalog team does a maintenance and curation of the data.
Mhm.
But you can find, of course, some sometimes it will not be very consistent, but overall it's very good quality. And so most of the time these results are backed by a publication. So if we want to try to find it, so in this they were saying, so the associations, the studies, and here we go, the journal, publication date. Here we have a link to the study. We have the sample size, the platform of SNPs.
So basically, it's disease association with the genes, correct?
Yes.
Okay.
So in on more the drug discovery side, we could imagine more molecular and protein based association scores to find proteins that are involved in the same pathways, drugs that target the same diseases. In our other project, we have a data frame where we have, for one drug, the targeted gene. So these are the kind of data sets you can work on also and visualize as a graph. The SNP data will be more on the research side maybe. And so for drug discovery, you would be more interested in molecular profiles.
And the molecular profiles would be like specific to each organization and like.
So it could be, for example, the affinity between two compounds, or like two antibodies. Are they targeted by the same drug? So it's not really my field of expertise. Um, but one example is another lab in our department has done a foundational model, an LLM trained only on genetic sequences, protein-protein interactions. And so with these kind of LLM models, you could have embedding models similar to what you have with Word2Vec, or, and so you would give, for example, the name of a protein. It would give you an embedding dimension of, for example, 1000 scores for each of these dimensions. And you could give it a second molecular component, and you could do a cosine similarity between the two. So I was hoping to be able to show you a data frame a bit like this in this workshop, but I had to only focus on SNP and open access publication data that we'll see in part 2. But in the close future, I hope to maybe do a new vignette with this foundational model they developed.
Thank you so much. Appreciate it.
So in the vignette, we also have, coming back to the previous data set, suicidality. Here we have a bit the same behavior with a lot of other, the group other has a lot of elements. So this could be refined. Maybe we could take out all those genes because the visualization isn't so great with them. So one thing we can see in this one, so it's clearer on a bright theme, but even on the dark theme, you can see it a bit. Some edges have different colors, and the different color indicates that these nodes are connected to both target nodes. And these ones, even though they're in between, they are only connected to one. So these are the kind of nodes you will want to focus on. And so we can see, okay, this one, the group is NLGN1. This one, the group is this guy, C15. And this is a bit how we use it in our research. Before concluding this part, I give a bit more details about the groupings, and I talk about so there's the two cases, the floating grouping and the anchored grouping. And the vignette only shows one type, and this is the other type that I didn't incorporate in the vignette. I still had some improvements to, I wasn't sure about what example data set to use. And so, yeah, to repeat what I said at the beginning, the big difference is that in here you have the group, and when you click on it, you have the children node. So two different ways of organizing it. In the hospital setting, it's interesting when we have, for example, different identifiers for lab tests, but that are essentially the same lab test. And so that's how we use it. We just look at the group. So for example, here the lab tests. So these guys have only one children, so it's not the best examples, but that's the idea. We could have one type of analysis and different ways the different hospitals, across hospitals, how they performed it. It also reduces the number of nodes overall, so it can make it a bit easier to read. So I'm going to take a little 5-minute break, and we'll start the second part, where we will focus more on embedding metrics and with an example with open access publications. So as I'm describing in the vignette, previously we had a data frame where we had all the relationships defined. And in our lab, what we often do is that, so we have all our electronic health records. We want to draw a knowledge graph based on that. So we have to determine what are the significant associations. And so in GWAS, we would do the p-value below 10^-8, for example. And in NLP, natural language processing, maybe I'll come back to the question a bit later. Can we clean the graph object? I'm not sure about the question. You can try to rephrase. So I was saying that we want to determine a significance threshold based on our pairwise cosine similarity matrix between our concepts. So our pairwise cosine similarity matrix can be derived either via Word2Vec or LLMs. I talk about this in this GitLab repo. I explain a bit how I did it with some open access publications. So here is a bit of code to download the publications. We do a bit of text pre-processing. We put everything to lowercase, remove some punctuation. And in this function, NLP embeds is mainly a wrapper to the text2vec R package, which performs Word2Vec with glove word embeddings. So the basic idea is that you have a sliding window over your text. And so the sliding window can be 20 words. And each time 2 words appear in the same window, they are a bit more correlated. So at the end, you will have in rows each of your concepts. So it can be each of your word, or you can pre-process your text data set to focus only on medical concepts. So in rows, you have your concepts, and in columns, you have the dimension of your word2vec algorithm. In this example, it was 100. And with this, we can do a cosine similarity between every element. And so we get a pairwise symmetric matrix with of our concepts. And so their similarity within the 100 dimensions. So we can use cosine similarity. We can also use inner product. In this example, I show a bit how we can explore such embeddings. We can type in some words. They will give us the 50 closest words. So the 50 that appeared the most frequently in the same sliding window. Here is a bit a novel method I developed that I'm not going to explicit too much here. It relies on the well-known operation in word2vec results. For example, if we do Paris minus France plus Germany, the closest match would be Berlin. And so it shows that we can learn some concepts of capitals, and it also shows that we can do arithmetic operations on our concepts. And so I use these arithmetic operations to explore my embeddings and find groups of concepts. That's basically what I'm explaining here. But in this part, evaluation is what we're a bit more interested in. So we'll come back to this. So we were saying we have a pairwise similarity matrix. And so let's say we have 10,000 concepts. If we want to focus on one concept, we're not going to graph this 10,000. We want to find a threshold. So without any further information, what we can do is take 5%. We determine the global threshold on our whole matrix. So all the cosine similarities, we take 5%. And so we take the quantile and we threshold. So more like, depending on how you look at it, 5% or 95%. And so for each concept, all the concepts that have a similarity higher than that, we will graph them as our knowledge graph. And so this can be a bit tricky to do with a knowledge graph, because you're not going to be able to do your pairwise similarity matrix if you have more than 10,000 elements, basically, because you will need a huge RAM. And so that's one of the cool features of the Kgraph package, is that it will do that computation on the fly. And so it enables you to basically have as much concepts as you want, and it will just compute the closest ones as you ask for the knowledge graph of that concept. So let's have a look at how is it. So in the vignette, I first explain a bit all this, and I start with the example. So in the package submitted on CRAN, I took a small subset of the larger 10,000 embeddings I developed for that project. So I was doing the analysis on 2,000 open access publications on the full text, and I I had around 30,000 unique words slash concepts, and in the package on CRAN, there's a data set with 3,000. So it's a pairwise similarity matrix of 3,000 concepts, or 1,000 maybe. So this is it's already loaded in my session because I rendered the vignette already. So you can load it with the data call. Oh yeah, so this is not, sorry, this is not a cosine pairwise similarity matrix, it's the embedding matrix. So we have 1,000 words and there are 100 dimensions in Word2Vec. And the pairwise similarity matrix will be computed by the K-graph functions. So to avoid needing too much RAM, there's a parameter in this function max concepts. And so this will be, the more you have RAM, the higher it can be, basically. If your number of concepts is above that, it will do on-the-fly calculation. And if it's below, it will just compute the full. If you have more, it will just take a sample. So if you do several runs, you may have slightly different results. But it should be very, so you can increase the max concepts to have more stable results. So let's have a look at this call. Or maybe first we'll have a quick look at our embedding matrix. So these are concepts identifiers from the UMLS database, so Unified Medical Language System, maybe. And so it's medical terms. And I have the dictionary, the associated dictionary. So in my dictionary, it would look like this. Here we have the identifier and the descriptions. So tumor, prostate, etc. UMLS also provides a group information, so you know a bit the pathway or what kind of concept it refers to. So this is our embedding matrix. Each of the column corresponds to the Word2Vec dimension. We have basically a score between -1 and 1. So we're going to call the fit embeds kg function on that. First parameter is the embedding matrix. Second parameter is the distance. So cosine, or I think it's inprod for inner product, which is detailed more in the function. We can have a look at the function documentation. Here we have the different similarities we can use. So our main interest in this object will be the threshold that we determined. So this is the 5% quantile that will basically all the similarities above this will be considered significantly similar. And the function also has the threshold proj parameter with which you can define yourself the quantile you want. So what we often do is that, so there's the default of 005, and that is always returned, and there is the one that you specify that is returned. And I added the 005 as a default because one usual application can be, we're not really sure if we want to use 5% or 10%, so we can compare the results between the both. So with this call with 0.9, we basically have both thresholds. We have the 0.9 and the 0.95. So with this, we determine our threshold. Talked about the dictionary. We want to identify which nodes we want to focus on. So we're going to search for the string in our dictionary descriptions. We're just going to take the two first occurrences, and we're going to retrieve the ID. So these are the two IDs we are going to focus on, that we are going to call with the K-graph functions. So in this one, I call the build K-graph from fit because I already computed the fit, but we can also call build K-graph directly on the embedding matrix, which will compute the fit and the threshold by itself. So this returns us a K-graph object that we can plot in a similar way as previously. So I showed it a bit in the beginning. Here we have our two target nodes. These are the group nodes. On this side, more mental disorders. On this side, more clinical disease. So the bigger ones are the most associated, it's the ones we want to check in the first. And a few other interesting group nodes. So finding, it can be laboratory tests or psychology questionnaires. And so as I was saying previously, the dark edges here show the nodes that are shared between the two. So these are the ones we want to focus particularly on. We can have spurious correlations, but we might be able to find a pattern. And so clicking on them will show the group node to which they are related. This is the finding. This one is a group node a bit further away. We can click on the group node to see only those. Going to check a bit the chat. So for the question about maybe focusing on a specific group, what you could do is subset your embedding matrix, but there is no way to do it directly from the K-graph functions. So here I showed how we were able to produce the knowledge graph based only on the embedding matrix. But what most of the time what we will have is actually a paired data frame that will contain known pairs of concepts. So, for example, we know that schizophrenia is a mental health disease. We know that this gene is associated with this disease. That's the kind of relations. So we know some of that. It can be curated by clinicians. We can derive them from GWAS summary results, or so we have these known pairs, and what we're thinking is that these are the things we already know, but maybe with our knowledge graph we can find novel associations. And so instead of just using a 5% quantile, we're going to do a 5% false positive. So it's not sure if I'm going to be able to explain this easily, but let's go back to this GitHub repository. So I'm giving the example of the PrimeKG database, which is developed by the lab I told you about that did the foundation model of molecular profiles, Professor Zitnik. And so the PrimeKG database, it compiles some disease-disease relations and disease-drug relations. And these are what we're particularly going to use. But the PrimeKG database also is more focused on bioinformatics drug discovery. And there's a lot of information about genetic pathways and protein interactions. This paper can be very useful. It's a bit how it was written by my lab before I arrived, and it helped me a lot understand this 5% false positive. The idea is that, let's say we have our cosine similarity threshold, and we have the concept we want to graph. Let's imagine we have 100 concepts that are returned. With our known pairs, we can check how much are true and how much are false. The false ones will be random, just random combinations of our concepts. Since we have a large number of concepts, the number of related concepts should be much smaller. And so we can assume that taking random will most of the time be true negatives. And so I was saying we have one concept, it returns 100 related concepts. And what we're going to look at is how much of the true negatives appear in these 100. And this will give us the percent of false positive, because we saw it's false positives because we saw that they were related, because it's the 100 most similar. But we actually have in our data, in our known pairs, that they are not. So the number of false positives is the number of true negatives that appear in our 100. So this enables us to determine, let me take a step back. So let's say that out of our 100, if we have 5 true negatives, then it will be what exactly the 5% false positive. If we have 10 true negatives, it would be a 10% false positive. And that's the threshold we're trying to determine. And so if we have 10 true negatives in our 100, we will reduce the 100 down to having only 5 true negatives. And once we have that, it's our 5% false positive threshold. So here I was giving the example for just one concept, but we basically do that for the whole matrix. And either the whole matrix or a sample if the number of concepts is too large and that we can't do the full matrix. So this is how we determine a 5% false positive using known pairs. And we can also compute the AUC performance score with these known pairs. And so this, now that I've explained all this, we can move on to the second part, last part of the vignette. So it's basically what I said, much shorter. I tried to explain it in more details here. So in the KGraph package, there's a subset of the prime KG database that we can use. So let's load it up, see what it looks like. So we have our first concept, second concept, and descriptions. So we can manually curate it. So here, like just disease or disorder. So as I was saying, the Zitnik lab is a bit more focused on bioinformatics, drug discovery, and our lab is a bit more focused on hospitals. We have a similar known pairs data frame that is not public. So these relations may seem a bit trivial, but you can also do more. If we take the tail, here we have, for example, heart disease, heart conduction disease. digestive system cancer, malignant neoplasm. So this starts to be a bit more interesting relations. So this is so you can create your known pairs as long as they have the same concept names as you use in your embedding and your dictionary. And the K-graph package will do your 5% false positive and your AUC score. So this time we recall the same function fit embeds kg as previously, and we add the pairs parameter, and we select the relevant columns, so 1 and 3, the two IDs. This time the return object is slightly different. There's basically a bit more fields. There's both objects are enough to show the graph, but in this one we can show AUC, and so it's in the ROC object. It's computed with PROC package, and here we have our result, AUC 0.62. And so how we use this in practice is that, for example, the word2vec dimension was 100, and my latest results, so I'm more doing PMI SVD, pointwise mutual information, singular value decomposition. But it's very similar to word2vec nonetheless. And what I've seen is that we need a much higher dimension. And my optimal results are more with a 2000 embedding dimension. And I get AUCs up to 0.75. So quite a large increase. And it's understandable because the natural language processing relationships are very diverse. And that's a bit why we need much more dimensions to understand them correctly. With a 100 dimension, you can do satisfying results for codified data from electronic health records. So codified data will be diagnosis, medications prescribed, lab tests, procedures, such as surgery. But when you start doing NLP, you have much more various relationships, and so you need a much higher dimension to understand them correctly. That's at least what my results point to. And so this ROC object can be plotted with the PROC function. So for those who don't really know about AUC curves, we're mostly interested in this region, let's say. The straight line would be a AUC of 0.5, which would correspond to a random prediction. So 0.6 is slightly better than random prediction. It's not great, but it's still significantly better. And I was talking before about 5% false positive. So it would correspond to a 0.95 specificity. And so it would be basically around here, the cutoff. And so we would have, so it's not directly related to AUC in the sense that it's just, AUC is more global. But when we're trying to do drug, for example, in drug discovery or in the medical setting, when we're trying to predict new relationships between disease and drugs or between medical concepts, we're actually not going to look at the whole predictions. We're just going to look at the 5% false positive. So to have numeric values for that, we can also use PPV, positive predictive value. But so I won't give more details about that. But so one of the interesting thing is the shape of the AUC curve, and we can see that here it's not symmetric. It's, can have a look at the figure from the vignette. So I was saying that it's not symmetric. We can see that the area above the random line is larger at the beginning than at the end. And so I'm not going to explain all the implications of that, but the shape of your AUC curve may be one of the thing you take into account when you're comparing prediction models. Basically, for the same AUC score, you would prefer an AUC that is skewed like this rather than an AUC that is symmetric, because it will mean that your 5% false positive will be better. Your PPV will be better at 5% false positive. So for this one, I don't really have another data set to show. I just uploaded the full data set, so we can try to have a look at that. So I can try to do that, and if you have any questions. So here I loaded the full dataset file of 25 megabytes. So we can see that we have 30,000 concepts compared to the 1,000 previously. And we can do the same call. So we could also use a expanded version of the pairs dataset. Here we have a much lower AUC. One of the reasons is that in this analysis, I didn't subset on frequency terms. So the words that appeared in my text corpus very rarely are maybe often misclassified because there was only one or two occurrences that enabled to determine the word2vec embedding. And so one of the things you can do that would improve your area under the curve is to subset to only terms that appear on 10 times. And most of the time, as you increase this threshold, so you decrease your number of concepts, obviously, but you increase your AUC as well. And in one of our projects, we are trying to define a number of related concepts to suicidality. And so we have to balance a bit these two parameters the minimum frequency and the AUC resulting to get the most appropriate sets of terms that will be useful for downstream analysis and suicide risk prediction. We can have a look at the other distances here in the product. Slightly better AUC. From my experiments, it's usually cosine better, but it can vary a bit. Of course, as I was saying, there's a bit of sampling going on, so you might want to bootstrap a bit also. Here we do another time cosine, we have 56 compared to 54 before. So these AUCs are very low, but still it shows that you have a bit of a confidence interval to take into account. So we can try to graph this object. So when I did the subset, I selected it to be the concepts that were important for these specific concepts. So that's why I think that oh no, I didn't refresh my KG object. So that's why. So once we recall the fit, we call the build kgraph from fit function. We pass it the embedding matrix because we have to do on the fly computation of the cosine similarities. Yeah, so here, since it's also the dictionary, I subsetted for the public package. So the kgraph function is giving us an alert that our dictionary is not complete. There are identifiers that we're trying to graph that are not found in the dictionary. So this should be this could be a stop, but it's just a warning or a message. So this is what our new graph looks like. So this other group, I'm working a bit on it. Maybe try to approach it in another way. This graph doesn't look great. So here, typically, we would want to have a bit more stringent threshold. So we can, we were using yeah, so, oh yeah, I understand now. The other group is so big because our dictionary is incomplete. And all these yellow guys are all the guys that we don't have descriptions for. So this is why we still have their identifier C074. And this node is an example of a concept we had already in our dictionary. So maybe we'll just come back to and that's pretty much it. Thank you all for joining, and I'm happy to answer any questions that you still have.
So the dictionary, did you get it from the EMR data?
From the UMLS.
I see. So if you, I mean, your analysis is based on EMR data too, correct?
Yes.
So yeah, that's like a roadblock for people like us who want to do something. Yeah.
So the analysis I present in the vignette is done on open access publications. The EMR data is what we do internally in our lab, but the vignette shows on open access publications.
Okay. Thanks.
And you can reproduce that with this repository. And so one interesting exercise could be to try to increase the number of Word2Vec dimensions. And so this would be around here with the str2vectors function. So I can't show you that function in the POSIT cloud because it's a package I didn't submit on CRAN yet, but it is in the Docker image, and the GitLab repo is also public. And so in this function, you have a parameter to which you can set the embedding dimension. And in this, so here what I did was that I downloaded open access publications from Europe PNC FTP. So you can either access them with these R packages directly, but if you want to do your analysis on many records, you might want to download in bulk rather than via the API. So that's why I downloaded from the FTP. And so what I did was that locally, I would select all the publications that were talking about suicide, and I built a new XML file containing only those articles. And so this can be done in a similar way we could search just for mental health, or we could search for the name of a disease, rheumatoid arthritis. So the first step would be to download the open access publications. Second step, to build a new XML file. And after that, read the XML file with the tidy PMC package functions or Europe PMC, and start doing text processing and calling glove Word2Vec. And once you have your Word2Vec matrix, you can use the kgraph package. But thank you all for the nice words in the comments. It was great to share some time with you.